Hello, welcome back. So in this third class on structural bioinformatics, we'll be talking about two topics, one that relates to predicting protein structures from sequences, and the other about predicting the impact of mutations. So this video gives you some introductory material about these topics. I'm going to start about describing generally the folding problem and some of the key ideas that have helped to make predictions about structures from sequences. So in principle, all the information that relates to the three-dimensional structure of a protein should be encoded in the sequence. And so the residues will have different propensities to organize themselves sequentially into patterns that relate to the secondary structures, such as alpha helixes and beta sheets. And then in principle, the different organizations of these alpha helixes and beta sheets should in principle have the information required to fold a protein into the three-dimensional shape that it has. And one of the ways that people try to predict structures from sequences is simply by homology, as we discussed before. If two structures have sequences that are over 30% sequence identity, then by and large, they will typically adopt the same structures or very similar structures. So the higher the sequence identities, then the higher success in building a correct prediction by homology. So if one cannot do that because maybe there isn't a structure already known that has a very sufficiently high sequence identity, or if you really care about the details, so these homology models may be generally correct overall, fairly correct, but then still fail in details. You would need better ways of making these predictions. Well, it's actually quite hard to do it from just a sequence alone. And the problem is really the number of possibilities in which the side chains and all the atoms may be positioned. So there's a lot of degrees of freedom. So that's been described as the Leventile's paradox. So Leventile estimated that there could be over 10 to the power 300 possible conformations in a typical protein sequences to fold into a three-dimensional structure. And so it would take more than the time of the age of the universe to find the correct conformation if it was done purely by random search. Yet, inside cells, proteins fold in milliseconds. So it's clear that this search for the corrected folded state cannot occur purely by random search. There must be some local terms, sort of local interactions that sort of drive an unrandom search to the outcome. So this idea that there is a landscape, and so some states are more probable than others, and so there must be a guided search to the ultimate outcome of the folded state. And so in principle, it should be guided by these local interactions, like these local propensities for secondary structural elements, and then giving rise to a higher order, a bigger structure. So how can one predict structures from sequence alone? If we think about these ideas that I've just described, then two elements that we need. One would be a way of scoring how correct, how stable a given structure is from a given sequence. And then a very fast way to sample possibilities. So you ideally would like to sample through the space of possibilities in an unrandom fashion, such that you would go towards the most stable states. And of course, you maybe have a problem where you may be locally stuck in a minimum. And so you also need ways of probing away from those local minimum. So what are some ideas that have helped in performing predictions of structures from sequence? One is simply by homology, as I just described. And the other thing is not all possible conformations exist in nature. So there must be also ways of just learning from existing structures where our conformations are simply impossible, and therefore there's no point in searching through those conformations. And the other very key idea is that evolution, in particular co-evolution, provides information about the three-dimensional shape of the protein. And that has been a very key idea in developing very, very good structure predictors. And so we discussed this briefly in the past, but there's this idea that if we take a protein sequence and we build an alignment across many other homologous sequences in other species, in principle, it's been shown, in fact, that positions that are in close contact in three-dimensional shapes proteins will tend to co-evolve, which are shown here as in examples in this sort of toy alignments. And so in principle, it should be possible to extract that information from the alignments in order to help predict the structures of proteins. So this has been one sort of ideas has been first a long time ago. One would take a sequence, build a multiple sequence alignment, and then from that multiple sequence alignment, we would get some information about positions that are co-evolving or conserved, and then from then predict, for example, secondary structural elements. And as I said, we could take information about the structures that exist in the PTP, the ones that have been experimentally solved already, to get an understanding about the different types of fragments, so parts of proteins that would be consistent with the secondary structural information or other types of information one would collect already from the sequence or the alignment, and then have a way of using those local fragments to build possible bigger proteins and some way of measuring the energy of those different possibilities. So this would be a faster non-random search space of possibilities. And then there were some improvements on this idea, which is directly trying to have some machine learning methods. I'm not going to go into the details of this, where you would try to predict the three-dimensional contacts. So essentially the residues in 3 dimensions that would be close together in 3D directly from the multiple sequence alignment. And that is a very important idea, because then this idea was the idea that developed the most recent methods. And so then from then on, what has happened is more of these parts of this approach have been taken over by neural network models. Again, without going into many details, now the idea progressed towards increasingly adding neural networks to different aspects of the problem of going from the multiple sequence alignment to the prediction of the structure. And so AlphaFold 2, which was the most recent development in Rosetta Fold, and there have been a few others that have been released, again, take this multiple sequence alignment, taking this neural network models are able to very accurately predict the three-dimensional distance constraints of pairs of residues, and then have another neural network model that helps take in these matrices of pairwise distances estimates to predict the structures. And so this has led to what could be considered a revolution, whereby over the years, you can see this is an experiment called the CASP competition, where experimental structures that have not yet been released to the public are given to predictors. So every 2 years there's a competition where people try to predict structures based on the sequence. And AlphaFold, in particular in 2020, has released predictions that are reaching a level of performance that it can be considered as good as experimental models, which is only true for certain types of structures. So it only is true for proteins that are fairly well globular and organized, and also potentially not membrane bound and so on. So there's some constraints about which ones can be used, but it's been a really big improvement in the capacity to predict structure from sequence. Here's just 2 examples. Of course, these are examples that obviously would look good. So the experimental result is in green and the computational prediction is blue. And there's almost like a perfect overlap between these 2. And so you don't necessarily have to perform these predictions yourself now. So there are tools and databases on which you can retrieve either homology models in the Swiss model database or AlphaFold predicted structures from the AlphaFold database. And we're in class going to look through these and just have an understanding of what these are and what they hold, and some information about the quality criteria that you have to look at if you are going to use one of these models. So in particular for AlphaFold, there are 2 confidence estimates that you should have a look at and understand. And we're going to go through these in class. One is the local confidence of estimated quality for each residue, which is this PLDT, and you can have an average across the whole protein as well. And then there's also this predicted line error, which is the confidence in the relative position between 2 residues or pairs of residues. And again, we'll go over these estimates throughout the class. You can make your own predictions easily as well if you wanted to. So there's a online website where you could do this. And in this case, it's through a Colab notebook. And we're going to go through in class one of these examples. So the second aspect that we're going to cover in class is once you have a structure, one very important application of this is to predict the impact of mutations. So if I have a protein structure, I can, in the computer, make a missense mutation, change the amino acid, and try to ask, does this mutation have an impact on, for example, the stability of a protein? So there are many ways of predicting the impact of mutations. I should say upfront that the most accurate ways are still using protein alignments. So protein structures can give you very correct predictions of the impact of mutations, but using protein alignments can give you even more accurate predictions. So essentially, the key idea here would be that if I have a very conserved position in the alignment, if I rarely see the mutation that I'm trying to predict, then this is very unlikely that this mutation will be tolerated in the protein. And of course, the deeper the alignment, the more accurate this prediction would make. And there are many sequence-based methods to predict the impact of mutations. We're not going to cover these. So here, instead, we're going to look at mutations that have to do with predicting impact of mutations using structure. And of course, you can already imagine if you were to predict, for example, impact of mutation in the core of a protein, these are very likely to be destabilizing, while if you're predicting the impact of mutation at the surface of a protein, then these are less likely to be destabilizing. And so, as I said, there are computational methods that can help us to estimate the stability of a protein structure based on all the energy terms that go into, for example, predicting whether a hydrogen bond would be favorable, whether, for example, if different residues are clashing with each other, that would be not very favorable. And so this delta G, this change in Gibbs free energy, should be negative when the folded state is very stable. And so if we can predict this delta G, which is essentially giving you a measure of stability, we should also be able to quantify or computationally measure the impact of a mutation on the change in this delta G. So this would be the delta G. And in this case, if the delta G is bigger than 0, then this mutation is destabilizing. If the delta G is negative, lower than 0, then this would be a stabilizing mutations. And on the graph, you can see some predicted differences in stability, so this delta G, for pathogenic mutations, neutral mutations, and mutations that occur in the population. So these 1,000 genome variants should not be very deleterious on average. And so you can see that the pathogenic mutations, which is the dark line, tend to be shifted towards the right. So they tend to be more positive, which is the expectation. So pathogenic mutations should be predicted to be more deleterious, so more positive. So oftentimes a threshold of about delta G of bigger than 2, which is equivalent to 2 kcal per mole, is considered to be potentially deleterious. And so in order to make these predictions, there will be different tools that can do this. In this case, we're going to use FoldX. So FoldX is a computational method that is not a trained predictor. So it really has a series of energy terms that try to quantify the impact of a sterical clash, or when amino acids are overlapping with each other, or, for example, the positive and negative charge interactions, or hydrophobic effects, and so on. So all this information has different energy terms in FoldX, and so FoldX can open a structure and try to predict the impact of a given mutation on that structure.